Bismillahirrahmanirrahim. Hello, everyone. Our today's topic is pyrimidine metabolism. You may be knowing that purine and pyrimidine nucleotides are the components of nucleic acid, both RNA as well as the DNA. So, what we are going to do today is discuss about the biosynthesis, de novo biosynthesis and synthesis of pyrimidine nucleotides. Our today's point that we are discussing about, that is the learning objectives of today's lecture, are nucleosides, nucleotides, and their function in DNA and RNA. That is, we will differentiate between nucleosides and nucleotides and what their kind of a role they play in nucleic acids like DNA as well as in RNA. Then we'll also discuss about the structure and function of pyrimidines. Then, when the pyrimidine ring is synthesized, then we'll discuss the origin of different atoms in the pyrimidine ring. That is, what atoms contribute during the synthesis of pyrimidine ring. Then after this, we'll be discussing about essential features of pyrimidine synthesis and their catabolism. And finally, we'll discuss about the clinical aspect of pyrimidine metabolism and their disorders. Starting with the structure or building block of the nitrogen bases, you may be recalling that there are two types of bases are present in nucleic acids. One type are purines, which are 9-membered heterocyclic ring structure containing molecule, and the other ones are pyrimidines, which is a 6-atom containing ring. So we have two types of purines, adenine and guanine. Both are present in DNA as well as in RNA. As far as the pyrimidine is concerned, pyrimidines are of three types. One is cytosine, which is present in both in RNA as well as in DNA. Then the other one is thymine, present only in DNA. And the third one is uracil, which is present in RNA. Nitrogen-containing bases, as I pointed out, are purines and pyrimidines. There are two purines, which are right here. You can see right here is the adenine, and right here is the guanine. And the pyrimidine bases are right here is the thymine, the cytosine, and uracil. Recall that thymine is present in DNA. Cytosine is present both in DNA as well as in RNA, and uracil is present only in RNA. As far as the nomenclature of nucleotides are concerned, nitrogen bases, look right here, adenine and guanine. When they are combined with the sugar, then they make the nucleoside. And the name of the nucleoside of purine bases ends with O-S-I-N-E, O-S-I-N-E. For adenine base, the nucleoside is adenosine. For guanine, it is guanosine. And if we combine phosphate with nucleoside, that is adenosine, then it becomes the nucleotide adenosine monophosphate. That is, depending upon the number of phosphate groups, we can make adenosine monophosphate for 2-adenosine diphosphate, and for 3-adenosine triphosphate. Similarly, guanosine combines with the phosphate, makes guanosine monophosphate, diphosphate, and guanosine triphosphate. The nomenclature of pyrimidines is slightly different because you can see the name of pyrimidines, for example, cytosine, it ends with OSINE. So therefore, in order to differentiate from pyrimidines, purines, then the nucleosides and nucleotide of the pyrimidines normally end with DINE. DINE. For cytosine, when combined with the sugar, makes a nucleoside, and the nucleoside of cytosine is known as cytidine. Then with the cytidine, if we combine one phosphate group, phosphate molecule, it becomes cytidine monophosphate or CMP. Combination with two phosphates, UDP, and three phosphates with CTP. For thymine, combination with sugar makes a nucleoside and known as thymidine. Thymidine. With one phosphate, thymidine monophosphate, two thymidine diphosphate, three thymidine triphosphate. These are the nucleotides. Uracil, when combined with the sugar, ribose, makes uridine. That is the nucleoside of uracil. When uridine nucleoside combines with phosphate group, it makes a nucleotide, uridine monophosphate or UMP. And for two phosphates, UDP, and three phosphates, UTP. As far as the functions of nucleotides are concerned, nucleotides play key roles in many cellular processes. So the functions of nucleotides are, number one, they are the activated precursors of RNA and DNA. That is, the activated purine and pyrimidine nucleotides are the raw material for the synthesis of RNA and DNA. That is, RNA and DNA both are synthesized from the activated purine and pyrimidine nucleotides. That is, having the triphosphates. That is ATP, GTP, UTP, CTP, and TTP. Adenine nucleotides are components of three major coenzymes. Because the coenzymes are the enzymes which work in collaboration with the enzyme and enhance the activity of that particular reaction. Adenine nucleotides like NAD, nicotinamide adenine dinucleotide, FAD, flavin adenine dinucleotide, and coenzyme A. These are the three coenzymes which are synthesized, which are part of the nucleotides. Nucleotide derivatives are activated intermediates in biosynthetic processes. That is, recall the synthesis of glycogen that we have already done. Nucleotide, the glycogen molecule is synthesized from glucose, but it is not directly from glucose, but activated form of glucose. In glycolytic pathway, the glucose is phosphorylated, converted into glucose 6-phosphate by hexokinase enzyme, and glucose 6-phosphate then goes to the glycolytic pathway. But for the synthesis of glycogen, glucose 6-phosphate is converted into glucose 1-phosphate first. Then it combines with the UDP and makes UDP glucose. UDP, that is uridine diphosphoglucose. This is the activated precursor for the synthesis of glycogen. Similarly, for the synthesis of fats, CTP glycerol. CTP glycerol is the precursor. And SAM, S-adenosyl methionine is the universal methyl group donor. Nucleotides serve as metabolic regulators as well. That is, they control the different metabolic pathways. For example, cyclic AMP, which is also known as second messenger, and the activation of cell signaling by these nucleotides. They also serve as a major currency of energy in the cell. ATP and GTP. Generally, ATP is considered as the major currency of energy in the cell, and also to some extent, GTP also serve as a major currency of energy in the cells. Many metabolic diseases have their etiology in nucleotide metabolism that we will be discussing in the later slides. As far as the biosynthesis of nucleotides are concerned, there are two pathways by which nucleotides are synthesized. One is de novo pathway or de novo synthesis, that is the synthesis of nucleotides by adding one by one atom from the scratch and keeps on building the nucleotide ring or nucleotide structure. The second pathway is salvage pathway. As you know, the salvage means the recycling of the molecules. So, salvage pathway means already synthesized pyrimidines or purines or their nucleosides then can be converted into nucleotides. So, de novo synthesis of pyrimidines leads to the synthesis of UMP first, and then from UMP we get the synthesis of deoxy-UMP and deoxy-TTP. From UMP, also we get the synthesis of UTP and CTP. So it means there are two pathways for the nucleotide biosynthesis, especially purines and pyrimidines. So because we are discussing pyrimidines, so de novo pathway and the salvage pathway. The difference between these two pathways is that de novo pathway requires much more energy for the synthesis as compared to salvage pathway, which requires less amount of energy. So biosynthesis of pyrimidines, as I discussed in the last slide, liver is the major site for the synthesis. There are two pathways, as we have discussed, de novo synthesis and salvage pathway. Both operate in cytoplasm. Biosynthesis of pyrimidine nucleotides, purine pyrimidine nucleosides, biosynthesis share common precursor. That is, for the synthesis of purine as well as pyrimidine nucleoside. Recall, nucleosides are the combination of purine or pyrimidines with sugar. So, for the biosynthesis of purine as well as the pyrimidine, we require, for both of them, we require a common precursor, which is PRPP, that is phosphoribosylpyrophosphate. pyrophosphate. And another common molecules are glutamine, aspartate, carbon dioxide, and for thymidylate, which is the one which is present in DNA, tetrahydrofolic acid, that is THF. Tetrahydrofolic acid is a derivative. The first enzyme, carbamoyl phosphate synthetase 2. is present in cytosol and used for pyrimidine synthesis. Let me refresh your memory. There are two types of carbamoyl phosphate synthetases. Carbamoyl phosphate synthetase 1 and carbamoyl phosphate synthetase 2. Carbamoyl phosphate synthetase 2 is present in cytosol, used for the synthesis of pyrimidines, and carbamoyl phosphate synthetase 1 is present in mitochondria and used for urea synthesis. We'll be discussing about these two enzymes in the later slides. In cytosol, for carbamoyl phosphate, for pyrimidine synthesis, glutamine is the nitrogen donor. And in mitochondria, for carbamoyl phosphate, for urea cycle, ammonia is the nitrogen donor. So this is the difference between these two that we'll be discussing in the later slides. The materials required for synthesis of pyrimidine, that what type of molecule, enzymes and other materials that we need for the synthesis of pyrimidines. Number one is the starting point is the synthesis of carbamoyl phosphate. Carbamoyl phosphate synthesized from carbon dioxide and glutamine in cytosol. The second molecule that we need is PRPP, that is phosphoribosyl pyrophosphate. More precisely, 5-phosphoribosyl 1-pyrophosphate. Then different type of enzymes which are involved in the pyrimidine biosynthesis is carbamoyl phosphate synthetase 2 that we have discussed in the last slide. Then another enzyme, transcarbamylase, dihydroorotase, dihydrogenase, transferase, and decarboxylase. These enzymes are involved during the biosynthesis of de novo biosynthesis of pyrimidines. Now, starting with the synthesis of pyrimidine. The synthesis of pyrimidine is much simpler. The pathway of synthesis of pyrimidine is much simpler as compared to purines. The major differences between the synthesis of purines and pyrimidines is that in case of purines, the purine ring is synthesized on phosphoribosyl pyrophosphate. That is, the first molecule is phosphoribosyl pyrophosphate, and on phosphoribosyl pyrophosphate, different atoms are added one by one sequentially, and we get the formation of purine nucleotide. In contrast to the pyrimidines, the first pyrimidine ring is synthesized from different atoms, and then once the ring is synthesized, then the phosphate sugar is added, that is the phosphoribosyl pyrophosphate is combined, and then we get the formation of nucleotide, pyrimidine nucleotide. So remember the difference between these two. Purines are synthesized on phosphoribosyl pyrophosphate by adding different atoms one by one. And finally, we get the purine nucleotide. In case of pyrimidines, the pyrimidine ring is first synthesized, and then after the synthesis, then phosphoribosyl pyrophosphate is added to make the nucleotide. Now, starting with the de novo synthesis of pyrimidine. Pyrimidine, the first reaction is carbon dioxide combination with glutamine in the presence of ATP. We get the formation of carbamoyl phosphate. You can see, look right here. ATP, carbon dioxide, glutamine, and carbamoyl phosphate synthetase 2 makes the carbamoyl phosphate, and we get the ADP and glutamate formation. Then carbamoyl phosphate combines with aspartate in the presence of an enzyme, aspartate transcarbamylase. In this case, the whole aspartic acid molecule combines with carbamoyl phosphate and makes carbamoyl aspartate. That is, aspartate plus carbamoyl phosphate makes carbamoyl aspartate, and the enzyme is aspartate transcarbamylase. Now, up to this point, all the atoms of the pyrimidine ring, are now completed. All we have to do is simply cyclize these atoms. So the next reaction is dihydroorotate, because the product formation is the dihydroorotic acid. That is the cyclization of the pyrimidine ring. So the first pyrimidine ring which is formed is dihydroorotate. And this dihydroorotate is reduced to orotate by the action of coenzyme FAD, which is reduced to FAD, converted to reduced form of FAD by the action of an enzyme dihydroorotate dehydrogenase. Look right here, dihydroorotate dehydrogenase. And we get the formation of orotate or orotic acid. Now, this is the complete ring of pyrimidine having extra carboxylic group. Then this combines with PRPP, phosphoribosyl pyrophosphate, in the presence of an enzyme, orotate phosphoribosyl transferase, and makes the orotidine monophosphate. Orotidine monophosphate, that is now what we have synthesized is the orotidine monophosphate, that is the nucleotide, OMP nucleotide. From this, the removal of this carbon dioxide present right here, you can see it, is removed by the enzyme OMP decarboxylase, and we get the formation of first pyrimidine nucleotide, that is UMP, uridine monophosphate. From this uridine monophosphate, the other nucleotides, pyrimidine nucleotide, that is cytidine monophosphate and thymidine monophosphate, are synthesized. Orotic aciduria and is a defect in the synthesis of pyrimidine that we will be discussing later on. Orotate, this is due to orotate phosphoribosyl transferase and OMP decarboxylase. They are separate domain of a single polypeptide UMP synthetase. Low activity of orotidine phosphate decarboxylase and orotate phosphoribosyl transferase both result in poor growth, megaloblastic anemia, and excretion of large amounts of orotic acid in urine, which is known as the orotic aciduria. The cure is, or improvement is, by the administration of uridine results in improvement of the anemia and decreased excretion of orotic acid. In mammalian cells, carbamoyl phosphate synthetase 2 is blocked or inhibited by UTP and activated by PRPP. In prokaryotic cells, aspartate transcarbamylase is inhibited by CTP and is the regulatory step. Now, because the first nucleotide which is synthesized de novo of pyrimidine was the UMP, now from UMP, then we can make the UTP by the action of an enzyme, UMP kinase, and ATP, which is converted into ADP. Recall, whenever phosphate is added to the substrate, the enzyme is kinase. So UMP by UMP kinase is converted into UDP. Now from this UDP, we can get the formation of CTP and deoxy-DMP. UDP, by the action of an enzyme, nucleoside diphosphate kinase, converted into UTP, and UTP is then converted into CTP by the addition of amino group contributed by glutamine, see right here, in the presence of ATP, and the enzyme CTP synthetase. We get the formation of CTP, and glutamine is converted into glutamate and plus ADP and inorganic phosphate. And we get the formation of CTP, which is pyrimidine present both in RNA as well as in DNA. Now from UDP, because the DMP is present in DNA, and DNA contains all the deoxynucleotides. So UDP is first reduced to deoxy-UDP, and the reducing agent is thyroidoxin, reduced form of thyroidoxin, which itself is oxidized, and it reduces the UDP into deoxy-UDP. That is reduction of the carbon number 2 of the ribose of nucleoside by the action of an enzyme, ribonucleotide reductase. Then deoxy-UDP is converted into deoxy-UMP, and deoxy-UMP is then converted into deoxy-DMP by the addition of methyl group donated by N5, N10 methylene tetrahydrofolic acid, which is converted into tetrahydrofolic acid. Now, when the pyrimidine ring is complete, then let's discuss the atoms that contribute in the molecules that contribute different atoms during the synthesis of pyrimidine ring. Look right here, the carbon number 2 and the nitrogen number 3 and of the pyrimidine ring is contributed by carbamoyl phosphate. That is nitrogen from glutamine and carbon from carbon dioxide. The rest of the 4 atoms, that is nitrogen number 1, carbon number 5, nitrogen number 1, carbon number 4, carbon number 5, and carbon number 6. These are contributed by aspartate. And sugar normally combines with the nitrogen number 1 of the pyrimidine ring. Orotic aciduria, as we have discussed earlier, it is due to inability of mitochondria due to damage to utilize carbamoyl phosphate. Recall, the carbamoyl phosphates are synthesized at 2 different locations of the cell. One is in mitochondria for urea cycle, and other one is cytosol for pyrimidine synthesis. And if there is a damage in the mitochondria, then the carbamoyl phosphate is available for cytosol or production of orotic acid, because the ultimate pyrimidine structure is the orotic acid, which is synthesized from the carbamoyl phosphate and the aspartate. Then excess amount of orotic acid is excreted in urine and known as results into orotic aciduria is due to deficiency of orotate phosphoribosyl transferase. If there is a deficiency of orotate phosphoribosyl transferase, then if the PRPP cannot combine with orotic acid, then excess of orotic acid is excreted in urine, known as the orotic aciduria. Now, deoxy-UMP, as we have discussed, in the presence of N5 and 10 methylene tetrahydrofolic acid, by the action of an enzyme, thymidylate synthetase, thymidylate synthase is converted into deoxy-DMP. Let me differentiate between synthase and synthetase. If during the synthetic reaction, if ATP is involved, then the enzyme is synthetase. And if there is no ATP is involved, then the enzyme is synthase. And in this case, you can see right here, there is no ATP is involved, and deoxy-UMP is converted into deoxy-TMP by the action of enzyme thymidylate synthase. Then the thymidylate, the dihydrofolate, which is formed after the donation of its methyl group, then is reduced to tetrahydrofolate in the presence of reduced NADPH by dihydrofolate reductase and regenerates tetrahydrofolate. The conversion of deoxy-UMP to deoxy-TMP is blocked or can be inhibited by 5-fluorodeoxy-UMP. So the 5-fluorodeoxy-UMP is the molecule that inhibits the conversion of deoxy-UMP to deoxy-TMP. UTP, which is synthesized, recall the first reaction. Complete pyrimidine is the UMP. From that, we can synthesize UTP by the addition of 2 phosphate molecules. And then UTP is converted into CTP. The difference between UTP and CTP is the presence of an additional amino group in the CTP. And that amino group is contributed by glutamine, which is converted into glutamate, and the enzyme is CTP synthetase because ATP is required in this reaction. Synthesis of CTP from UTP. Remember, the CTP required for RNA synthesis is converted to deoxy-CTP for DNA synthesis. This slide shows the difference between the 2 carbamoyl phosphate synthetases, that is synthetase 1 and carbamoyl phosphate synthetase 2. The cellular location of CPS1, that is carbamoyl phosphate synthetase 1, is mitochondria, and for carbamoyl phosphate synthetase 2 is the cytosol. Used for the pathway, carbamoyl phosphate synthetase 1 is used for urea cycle, and carbamoyl phosphate synthetase 2 is used for the pyrimidine synthesis. The source of nitrogen or ammonia, source of nitrogen for CPS1 is ammonia, and for CPS2, that is for pyrimidine, is amido group of glutamine. Amido group of glutamine. Regulators, CPS1 is activated by N-acetyl glutamate, and CPS2 is activated by PRPP and is inhibited, or inhibitor is UDP. So we have discussed so far the de novo pathway of pyrimidine synthesis. Now let's discuss salvage pathways of pyrimidine synthesis. Remember, the salvage means already synthesized base or the nucleoside. Uridine, which is a nucleoside, or cytidine, which is also a nucleoside, in the presence of ATP and by the action of enzyme, uridine and cytidine kinase is converted into UMP or CMP. That is what we have done. We already had uridine, that is nucleoside of uridine and cytidine. All we have to do is add ATP by the action of kinase, and they will be converted into UMP and CMP. And afterwards, we can convert them into UDP, CDP, and UTP and CTP. Thymidine, which is in the deoxy form, present in DNA, again by the action of thymidine kinase, which is the thymidine is the nucleoside, converted into deoxythymidine nucleotide. That is DTMP. Deoxycytidine, which is also component of the DNA, in the presence of ATP, converted into deoxy-CMP. Deoxycytidine kinase is the enzyme. Also, this phosphorylates deoxyguanine and deoxyadenine. Orotic acid, which is the first pyrimidine base which is formed, combines with the phosphorylate pyrophosphate, makes the OMP, that is orotate monophosphate, by the action of the enzyme orotate phosphoribosyl transferase. This is the first reaction. Orotic acid make the OMP. In this case, orotic acid is a simple base, pyrimidine derivative, and we are adding phosphoribosyl pyrophosphate, that is sugar in the presence of phosphate. So it will directly be converted into nucleotide rather than nucleoside. Catabolism of pyrimidines, cytosine, by the action of cytosine, deaminase is converted into uracil, and uracil then by the action of dihydrouracil dehydrogenase is reduced to 5,6-dihydrouracil. 5,6-dihydrouracil. Now this 5,6-dihydrouracil you can see right here, by the action is hydrolyzed by the action of an enzyme hydropyrimidine hydrase, converted into the 5,6-dihydrouracil, converted into beta-uridopropionic acid. Beta-uridopropionic acid then is further hydrolyzed, deaminated, and decarboxylated to form β-alanine by the action of an enzyme beta-uridopropionase, and we get the formation of β-alanine. 5-methylcytosine is deaminated by the enzyme deaminase and converted into thymine. Then, by the action of thymine dehydrogenase, right here, is reduced to 5,6-dihydrothymine. 5,6-dihydrothymine by the action of coenzyme NADPH, which is oxidized to NADP, and the thymine is reduced to 5,6-dihydrothymine. 5,6-dihydrothymine then is hydrolyzed by the action of an enzyme dihydrothymine hydrase, converted into beta-uridoisobutyrate. Beta-uridoisobutyrate. Beta-uridoisobutyrate, which is formed by the action of an enzyme by beta-uridoisobutyrase, is broken down into carbon dioxide, ammonia, and beta-aminoisobutyric acid. Beta-aminoisobutyric acid. Beta-aminoisobutyric acid, which is a degradative product of pyrimidines, is excreted in large quantities in leukemia after body is subjected to X-ray radiation. Recall that leukemia is the result of breakdown of the nucleotides. Pseudouridine, there is another nucleotide which is present in RNA, that is the especially in tRNA, no human enzyme catalyzes hydrolysis of phosphoride or phosphorylated of pseudouridine. Therefore, this base is excreted as such in urine of normal individuals. Thank you.